Mating between animals or plants that are closely related. Inbreeding. Inbreeding is when closely related individuals reproduce, which can increase the risk of genetic problems. Imagine you are trying to solve a jigsaw puzzle, but most of the pieces are from the same corner, making it hard to complete the entire picture. This is similar to inbreeding, where animals or plants that are closely related mate, which limits the genetic diversity needed for a healthy, complete gene pool. Just like using too many pieces from the same corner can lead to gaps and an incomplete puzzle, inbreeding can lead to a lack of genetic variation, resulting in health issues and vulnerabilities within a population, akin to leaving holes in the species' overall genetic puzzle. A genetically identical copy produced from a single cell. Clone. A clone is an exact copy of an organism created using its DNA. Imagine trying to replicate a favorite recipe that your grandmother used to make, ensuring every dish tastes exactly the same every time. This is similar to the concept of cloning, where a genetically identical copy is produced from a single cell, ensuring that the new organism is exactly like the original. Just as you carefully follow your grandmother's recipe to create an identical dish, cloning involves using the exact genetic instructions from a single cell to produce an identical organism. The process of changing the DNA of an organism. Genetic engineering. It's a technique used by scientists to directly alter the genes of an organism to achieve desired traits. Imagine trying to change the recipe of your favorite dish to make it healthier. This is similar to genetic engineering, where scientists alter the DNA of an organism to enhance or introduce desirable traits. Just as you might swap out ingredients in your recipe to reduce calories or add nutrients, genetic engineering involves swapping out sections of DNA to improve an organism's characteristics, such as making crops more resistant to pests or enhancing nutritional content. A small ring of DNA that can replicate independently of the chromosomes. Plasmid. A plasmid is a tiny circular piece of DNA found in bacteria that can copy itself and often carries useful genes, like those for antibiotic resistance. Imagine you're trying to get through a busy day, juggling multiple tasks at work, but you also have a personal project that you're passionate about and work on during breaks. This personal project is like a plasmid, a small ring of DNA within a bacterial cell that operates independently, just as your project runs alongside your main job responsibilities. Much like how your project doesn't interfere with your job, but can still progress independently, a plasmid can replicate on its own without relying on the main cellular processes linked to the chromosomes, allowing it to thrive in its own right. DNA created using fragments of DNA from different sources. Recombinant DNA. Recombinant DNA is a form of DNA that is engineered by combining genetic material from two or more different sources to create new genetic combinations. Imagine you're trying to make the perfect fruit salad, but you only have a few strawberries, a banana, and some grapes. Just like combining different fruits to create a delicious new dish, recombinant DNA involves stitching together fragments of DNA from different sources to create a new genetic sequence. In this analogy, each type of fruit you add represents a different DNA fragment from various sources, and the final fruit salad is akin to the recombinant DNA, offering new characteristics that the individual components couldn't achieve alone. An enzyme that cuts DNA at a specific nucleotide sequence. Restriction enzyme. A restriction enzyme acts like molecular scissors that identifies and slices DNA at specific spots. Imagine trying to find the right spot to cut a piece of wrapping paper to perfectly fit a gift. Just as you need to locate the correct line or pattern on the wrapping paper to make a precise cut, a restriction enzyme seeks out a specific sequence of nucleotides on a DNA strand to make its cut. In this analogy, The wrapping paper is like the DNA strand. The specific line or pattern is akin to the nucleotide sequence, and your scissors represent the restriction enzyme, emphasizing how both processes rely on precise identification to achieve the desired result. An organism that contains DNA from other organisms. Transgenic. Transgenic organisms have had genes from another species inserted into their DNA to give them new traits. Imagine trying to assemble a jigsaw puzzle, but you find that a few pieces are missing, so you borrow pieces from another puzzle to complete it. This situation is similar to transgenic organisms, where scientists integrate DNA from different species to fill in genetic gaps or add new traits. Just like using pieces from another puzzle to complete your picture, transgenic organisms incorporate foreign DNA to enhance or alter their genetic picture, resulting in new characteristics or abilities. The insertion of working copies of a gene into cells to correct genetic disorders. Gene therapy. It is a technique that uses healthy genes to replace or repair faulty ones to treat diseases. Imagine you have a broken zipper on your favorite jacket that keeps getting stuck. Fixing it permanently by replacing the faulty part with a new, functioning zipper is much like how gene therapy works. Gene therapy involves inserting working copies of a gene into cells to replace the faulty ones, just as you would replace the malfunctioning zipper to restore the jacket's function, ensuring smooth operation and eliminating the problem. The map of all the genes in a human cell. Human genome. The human genome is like an instruction manual containing all the genetic information needed to build and maintain a human being. Imagine trying to assemble a complex piece of IKEA furniture without any instructions. This challenge is similar to trying to understand how a human body is built and functions without a comprehensive guide. Just as the instruction manual provides a detailed plan to assemble every part of the furniture, the human genome is the complete map of all the genes in a human cell, guiding the formation and operation of our bodies. The breeding of plants or animals to produce desired traits. Selective breeding. Selective breeding is when humans choose certain plants or animals with specific traits to reproduce, so the offspring have those desired traits too. Imagine you're trying to organize your closet, but it's cluttered with clothes you rarely wear. Just as you'd sort through and keep only the clothes that fit well and suit your style, selective breeding involves choosing plants or animals with traits that are most desirable. In both cases, you're intentionally selecting the best options, whether it's clothes that match your needs or organisms with preferred traits, to create a more ideal collection, be it a wardrobe or a breed. A method of separating DNA strands by applying an electric current to a gel. Gel electrophoresis. It is a process that uses electricity to sort DNA fragments by size as they move through a gel. Imagine trying to sort different lengths of spaghetti noodles after a messy kitchen accident. Just like placing the noodles on a sloped surface allows gravity to pull the longer noodles further down than the shorter ones. Gel electrophoresis uses an electric current to pull DNA strands through a gel, with shorter strands moving faster and further than longer ones. In this analogy, the sloped surface is like the gel. The gravity is comparable to the electric current. And the different lengths of spaghetti represent the varied sizes of DNA strands being separated. A technique that makes copies of specific regions of DNA. Polymerase chain reaction. It is a method used to quickly make millions of exact copies of a particular DNA segment. Imagine you're trying to make enough cookies for a large party, but you only have a small batch of dough. Just like you need to keep doubling your cookie dough to get enough cookies for everyone, The polymerase chain reaction (PCR) technique amplifies specific regions of DNA to create enough copies for analysis. In this analogy, the small batch of dough represents the tiny amount of DNA you start with, and the repeated doubling process mirrors the PCR's cycles of replication, ensuring you have enough DNA to work with, just like you end up with enough cookies for your guests. The use of computer databases to store and analyze biological data. Bioinformatics. Bioinformatics is the science of using computers to understand and organize biological information. Imagine trying to organize and quickly find your favorite songs in an enormous music library. Much like how you create playlists and use search tools to efficiently manage and locate tracks, bioinformatics uses computer databases to store and analyze massive amounts of biological data. Just as playlists help you categorize and easily retrieve songs, bioinformatics databases categorize and analyze genetic information, allowing scientists to efficiently find and understand complex biological patterns. The identification of individuals from the unique sequence of their DNA. DNA fingerprinting. It's a scientific method used to identify people based on their unique DNA patterns. Imagine trying to identify your luggage at a crowded airport carousel, where every suitcase looks almost identical except for a few unique stickers or tags. Similarly, DNA fingerprinting identifies individuals by focusing on specific parts of their DNA that are unique, just like those distinguishing tags on your suitcase. Just as each suitcase might have a unique combination of stickers, each person has a unique sequence of DNA, allowing scientists to match or differentiate between individuals with precision. A glass slide or silicon chip with many DNA fragments. DNA microarray. It is a tool used to measure the activity of thousands of genes at once. Imagine trying to find the right key for one of many locks in your house. In a similar way, a DNA microarray acts like a set of keys, where each DNA fragment on the glass slide or silicon chip is a key designed to match a specific lock or gene sequence in a sample. Just as you would test each key until you find the one that fits the lock perfectly, scientists use DNA microarrays to match each DNA fragment with its corresponding gene sequence, allowing them to identify which genes are active in a sample. The study and comparison of genomes. Genomics. It is the branch of science that focuses on analyzing the complete set of DNA in organisms to understand their structure, function, and evolution. Imagine trying to organize a massive collection of jigsaw puzzles, each with thousands of pieces, and figuring out which pieces belong to which puzzle. This task is similar to genomics, where scientists study and compare entire genomes to understand how they fit together and differ from one another. Just as you would sort and examine each piece to identify patterns, colors, and shapes to assemble the correct puzzle, genomics involves identifying genes and genetic sequences to understand the structure and function of an organism's complete set of DNA. The study of proteins. Proteomics. Proteomics is the science of analyzing and understanding the structure and function of proteins in living organisms. Imagine trying to assemble a jigsaw puzzle with thousands of tiny pieces scattered all over your living room. Just as each piece of the puzzle has a unique shape and must fit perfectly into others to reveal the complete picture, proteins in our bodies have specific structures and functions that must work together precisely to sustain life. Proteomics, the study of proteins, is like gathering and understanding each piece of this biological puzzle, determining how they fit and function together to maintain the intricate image of life, much like solving the jigsaw puzzle to see the final picture. The use of living things to manufacture products. Biotechnology. It is a field of science that uses living organisms or systems to develop useful products and innovations. Imagine you're trying to bake a cake, but don't have all the ingredients at hand. Biotechnology is like having a magic kitchen, where instead of you doing all the work, you have tiny chefs, like yeast or bacteria, that can produce the ingredients you need. Just as you rely on these tiny chefs to whip up the components for your cake, biotechnology uses living organisms to produce necessary materials, like medicines or biofuels, by harnessing their natural abilities to synthesize complex compounds.